The image you are currently seeing is a cross-section of the cell during G2 interphase. The yellow cell membrane envelops the pale red center cytosol of the cell. Towards the left, there are two dark red centrosomes, which we will reference later. The blue nuclear membrane in the center contains the green chromatin and black nucleolus. During this phase, the cell prepares for division. The genetic information copied in S interphase is checked for accuracy, and organelle production and cell size increase. Following G2 interphase, the cell begins mitosis. During the first step, prophase, the nuclear membrane begins to break down and nucleolus completely dissociates. The newly compiled black chromosome is made up of two sister chromatids joined by blue centromeres. The centrosomes align themselves at opposite points of the cell, and the gray centrioles begin to form spindle fibers that will soon be used to orient the chromosomes. The next phase is called prometaphase. Spindle fibers from the centrosomes attach to the centromeres of the chromosomes. Notice how the kinetochore, the area where the spindle fibers attach to the centromere, has already been established before metaphase. Non-kinetochore microtubules are spindle fibers that have not attached to the chromosome and are used for later phases of mitosis. The next step is metaphase. During this phase, the kinetochore microtubules expand, causing the chromosomes to align along the metaphase plate, indicated by the green rectangle. This is a good checkpoint in the cell cycle. Everything is very simple from now on. Remember that the metaphase plate is an imaginary plane perpendicular to the spindle fibers. It is also important to note that the plate lies halfway between the two centrosomes. The next step, anaphase, is one of the final moments before the cell splits. As you can see, the centromere dismantles, breaking apart the two black daughter chromosomes. The kinetochore microtubules pull the genetic material towards the poles of the cell. Additionally, the poles of the cell move away from each other, causing the cell to elongate. Remember that the instant the kinetochore breaks from the metaphase plate region, the cell has begun anaphase. In telophase, the cell returns to its premitotic structure. As you can see, the spindle fibers, along with the centrosomes, have degenerated. The cleavage furrow is also evident. Notice the pent in the middle of the cell. The black nucleoli reform, as do the blue nuclear membranes. In the membrane, the condensed chromosomes now return back to chromatin. The key identifier for this phase is the cleavage furrow and the lack of microtubules. Finally, cytokinesis has occurred. How do we know this? There are two identical daughter cells. The cleavage furrow has successfully separated the two parts of the cell by a contractile ring made of actin and myosin. Thank you for watching.